Okay, perfect. Thank you, Paul and Selecta, for the kind invitation to be here today. My name is Matteo Marzi, and I'm from the Italian Institute of Technology, Milan. And today, I will be glad to share with you our experience with CRISPRi screenings to identify functional LncRNAs involved in adaptive responses in triple-negative breast tumors. So this is mainly the work of Bianca Giuliani, a PhD in our lab. And okay, even if this talk is sponsored by Selecta, the data I will show is 100% independent and unpublished research in our lab. So our lab has a long-lasting interest in triple-negative breast tumor, in particular in plasticity. So with adaptation of plasticity, we refer to the acquisition of the biological properties needed to survive in challenging environments. And triple-negative breast tumors are quite a good model to study adaptation because they display a strong phenotypic and transcriptional heterogeneity. This heterogeneity is fueled by either genetic or non-genetic events, which both contribute to tumor evolution. And this tumor evolution may be driven by pre-existing cancer stem cells in the tumor, or even by reprogramming of other cells. And reprogramming, for example, has been shown to be important in chemotherapy-treated patients, where when we observe a not complete pathological response, some cells may adapt and change their transcriptome. So the pathways involved in this kind of transcriptional plasticity has been quite well defined in the last few years. For example, partial EMT, hypoxia, inflammation, whatever. And our lab is an interest to study the connection of the non-coding components with this transcriptional response. So identify LncRNA modulators of adaptive properties and exploring their potential as therapeutic targets. So we have more than 18,000 annotated LncRNA in the genome, at least by gene code. Even if this annotation is actually not so precise, given the fact that LncRNA are very low in expression, and they also display a very specific cell type expression, specific expression. Furthermore, they also display a quite complex genomic organization. You may have intergenic LncRNAs with their own promoter, but you may also have a rather complex transcriptional unit where the LncRNAs share the promoter, for example, with a protein coding gene being transcribed in the opposite direction or in the same direction. So this may complicate a little bit the interpretation of CRISPR interference screens which target the DNA. So the association of LncRNA with cancer phenotype is actually pretty well established, but is limited to a few dozens of specific cases out of the 18,000 of LncRNA existing. So LncRNA may own either an oncogenic or a tumor suppressor function that has been implicated in many cancer phenotypes. And we are particularly interested in studying the possible regulatory networks that they can form with protein coding genes and DNA regulatory elements. This is illustrated in this example here for the MYC-PVT1 locus. PVT1 is a LncRNA at 50 kb of distance from MYC oncogene, and it has its own promoter. And when it is transcribed, it utilizes a set of enhancers in green here. But when the promoter of LncPVT1 is actually repressed, MYC is able to rewire the enhancers and use them for its own transcription. So this is enhancer competition, is a kind of an example of what we are looking for in our lab. So we have a lot of LncRNAs, and we want to screen those involved in adaptation. We decided to design 4 different screenings characterized by an increasing level of selective pressure. So the first one is a very easy proliferation screens for basic essential functions, cells grown in 2D monolayer. And then we perform a screen for 3D growth in methylcellulose. And these kind of spheroids are a proxy for stem cell properties of your cancer cells. We also perform a drug tolerance screen upon chemotherapy to identify the properties of the few colonies which emerge after the treatment. And finally, we perform an in vivo tumor initiation screen by orthotopically transplanting our cells in the mammary fat pad of mice. So given the fact that you have increased selective pressure, you will have fewer and fewer cells able to survive this kind of treatment. So maybe you can go genome-wide with a library in proliferation, but you have to scale down your library to the mid or even small scale for the other screens. So something that influences the screening scales is actually the model systems that you use. Because the more aggressive it is, the highest number of cells will be able to survive to treatment, and the largest the library you will be able to screen. So we obviously decided to focus on a very aggressive triple-negative breast cancer cell line, SUM159, that is able to form spheroids in 3D with a 20% of efficiency, which is actually sensitive to chemotherapy. We use the paclitaxel here, but a few cells are able to form drug-tolerant colonies like this. And finally, we tested by limiting dilution experiment the frequency of TAC in this line, which is quite high. 1 cell out of 700 is able to form tumors in vivo. So to focus on a set of meaningful candidates, we had to filter from the 18,000 lncRNA loci. And to do that, we used the guilty by association approach, which is based on the transcriptional characterization through RNA-seq of lncRNAs connected to contexts similar to those which we wanted to evaluate in the screenings. I mean by this that we, for example, profiled by RNA-seq the lncRNA induced after drug treatment of our cell lines and highly expressed in these drug-tolerant colonies. Or we use a different model system, the HMLI cell line, which contains a very tiny subpopulation of cells with cancer stem cell properties that can be FACS-sorted using the CD44 marker. So we FACS-sorted this population, and then we isolated the lncRNAs highly expressed in the cancer stem cell compartment. These are cell line. We also wanted to define lncRNA associated with primary tumors. So we got 30 tumors from the clinics, and we put shortly them in culture just to remove the stromal component, which can be a confounding factor when defining lncRNAs because they are very low in expression level. And then from these epithelial pure culture from primary tumors, we split them into groups according to the activity of the adaptation pathways that I told you at the beginning. And we isolated the lncRNA being connected with those cell lines having a very high activity of adaptive pathways. Finally, we also did the single-cell RNA sequencing of these models and the pickup candidates being expressed in a specific intracellular subpopulation, and a few candidates were selected from the literature already being implied in breast cancer biology. At this point, combining the information of all these models, we pick up 620 lncRNA candidates to be screened in the 4 conditions I already told you. We needed a potent manner to repress lncRNA expression, and we selected CRISPR interference because this system provides you with the possibility of switching off transcription of a locus without producing any genetic mutation in the DNA like the wild-type CRISPR. So we engineer our cell line with a piggyback transposable vector expressing in a doxycycline-inducible manner the dead Cas9 fused with the CRAB repressor domain. And then we tested several sgRNA to see how potent the system was. As you can see here, we could nicely knock down lncRNAs or protein coding genes almost to 99% efficiency. And we tested more than 50 guides before starting the library design, and we observed that in 70% of the cases, we were able to produce a knockdown of at least 80% of the target. So we selected the candidates, we selected the model system, and we selected the perturbation system. We were ready for the library design. So we got from Selecta this very nice construct depicted here, which not only expressed the sgRNA under the U6 promoter, but actually this cassette is embedded in the 3' UTR of a Pol2 transcript, quite close to the poly-A. So you can capture the sgRNA sequence by CROP-seq or perturb-seq, as we learned in the previous talk. So we decided to keep the library complexity small because, as I said, that these are very high-pressure screenings. So we designed a library of 3,400 sgRNA by just putting only 5 sgRNA for each candidate. We put a lot of non-targeting control, more than 200, and we also included a number of protein coding genes, many of them just being common essential genes to see if the library were working fine. And a couple of them, STAT3 and interleukin-6, which are required only for specific phenotypes, the 3D growth and in vivo, to probe the specificity of our analysis. Then we moved to the design of the sgRNA for link. As I said, the definition of the TSS for link is not very accurate, but we needed a very precise one because the CRISPRi activity window rely on a very tight window around the TSS. So to solve this problem, we used NetCage to precisely map the TSS for sgRNA, for lncRNAs. So we did this in collaboration with the Murakawa lab at the Riken Institute in the very same cell line that we used for the screen. And we got a very nice and precise NetCage peak for half of the candidate lncRNA of the library. In most cases, actually, the TSS was quite close to the annotated one in ensemble, in green here, so the distance is in the y-axis. But in 56 cases, they were quite far apart. So in these cases, the cage really impacted the sgRNA design. So I will now briefly focus on the data of 2 screens. The easiest one, the proliferation, and the hardest one, the TAC properties in vivo. So when you do a competition screen, you start by infecting your cell line with the library. And at this stage, you want to achieve a very balanced population expressing all sgRNA at the same level. Something that we nicely obtained, because as you can see here, 90% of the guides were expressed in a very tight 1.69 ratio. So a very balanced library that then is subjected to the selective pressure. And the selective pressure will lead some sgRNA to increase in frequency. You call this drop-in. And some other to decrease in frequency, and you call this dropout. The dropout sgRNA will target gene important for your specific phenotype. And you expect that the non-targeting control will maintain an equal frequency over time. So we did the 2D screen in 2 independent biological replicates, collecting the cells in doxycycline to induce the CRISPRi activity after 10, 17, and 24 days. And then we checked whether the representation of non-targeting control and the essential gene was as we expected. As you can see, comparing the endpoint with the starting point, we observed very stable representation of non-targeting controls in black, while SG for essential gene were lost in a very strong manner along our chain. So this is working. And what we wanted was to summarize the effects we got at gene level with a single score, summarizing all the time point and both the independent biological experiments. So we used for this purpose the MAGIC MLE framework, which at the end can provide you with a beta score, which is negative for dropouts and positive for drop-in. And here you have the ranked order from the gene with the lowest beta to the highest one. And as you can see, we nicely captured the genetic axis present between PVT1, which is the strongest drop-in of the screen, and MYC. As I want to remember, PVT1 repression will lead to MYC activation, and MYC is actually an important trigger for proliferation, of course. We also identify most of the essential gene as dropout, and also 24 lncRNAs with a statistically significant negative beta. So the 2D screen for us is just a reference to know which lncRNAs are actually associated with basic cellular function. So let's move to the in vivo screen. Here it was much more trickier. We had to orthotopically inject 500,000 cells with an intra-nipple injection into mammary fat pad of several independent mice. The SUM159 cells, with a latency of around 47 days, formed tumors with 100% of penetrance. And we collected sgRNA from 22 individual tumors separately. As expected, we observed here that the library coverage was dramatically affected by the strong selective pressure of the cells growing in vivo. So while the P0 on the left here was expressing all sgRNA more or less at the same level in blue, so a modest average level, in the tumor we observed that just 122 dominant clones emerged in each tumor, so each bar is a tumor on average. So we could detect just 50% of the library in each individual tumor. All the rest of the SG were zeros. So we had to change the analytical framework to be able to call hits in this condition. So for this purpose, we moved from MAGIC to a rank-based framework. And to do that, I decided to adapt the gene set enrichment analysis system, which is very well established, developing a sort of phenotype GSA. So briefly, it works by converting the abundance of sgRNA in each tumor, which is very variable because it's stochastic, into an ordered vector of ranking, where rank 1 will be the sgRNA with the highest frequency here, and at the bottom of the rank, you have all sgRNA with 0 frequency. Then we merged all the individual lists for the 22 tumors into a big list of ranking for all the experiment. And finally, we look at the position of sgRNA for each individual gene into this ordered global ranking. So for example, for MYC, you have 5 sgRNA in 22 tumors. So you have 110 different observations. And when you look where these observations are enriched, SG are strongly enriched at the bottom of the ranking, while PVT1 is strongly enriched at the top of the ranking, meaning that also in vivo, the MYC is acting as a dropout, and PVT1 is acting as a drop-in. And for many lncRNAs, like LASTER, which was published a few years ago to be important for breast cancer in vivo tumor formation, we observed this asymmetric distribution of sgRNA frequency. So even in this case, we recapitulated everything with a single score, the normalized enrichment score, which is actually provided directly by GSA framework. So the negative NAS will be dropouts, and the positive NAS will be drop-in, as before. So you observe here the position of MYC and PVT1, but I also want to stress that we got 13 protein coding genes to be important here. And look at the position of STAT3 in the 2T screening. It was in the middle without any effect, and it moved towards the top of dropout hits in vivo, meaning that we are capturing specificity. We identify here 45 lncRNAs statistically significant as dropouts, only 8 in common with 2D, meaning that many of them are important just in vivo. So to summarize, we successfully did all the screening. I have no time to go into the detail of each of them. And we identified it either with MAGIC or with the phenotype GSA framework. In total, in the first screening, we identified 75 lncRNA dropout, which might be lncRNA important for these specific phenotypes. I'm showing here the overlap of the hits coming from different screenings. So 12 hits are common to all the screens, 10 protein coding genes, and 2 lncRNAs. And these are basic functions, because probably they are just important for 2D proliferation. More importantly, we got 8 species in common between the 3D growth in vitro and in vivo, including STAT3 and interleukin-6 ST, so the positive controls, and 6 lncRNAs, which are very interesting for us. And finally, 29 hits in vivo, including LASTER and many others, were just specific for the in vivo setting. So we developed this reliable framework to study lncRNA plasticity by filtering through RNA-seq and then performing multiple competitive CRISPR screening. And I want to finish by saying that actually with CRISPR interference, we have identified important lncRNA loci. So it's important to understand that CRISPRi is targeting the DNA. So which is the real driver of the phenotype? The link, the possible close-by protein coding genes, some DNA element, and an answer. So we started mapping which hits do have a protein coding gene in the proximity, 39, but 36 do not have any proximal protein coding genes. And interestingly, here we found 2 microRNA clusters and a precursor for a circular RNA. So I can skip this, and I want to conclude by thanking again Bianca for the amazing work that she did with this screening, and my group in Milan, the Nicasio Lab, our funding agency, and all of you for being here at this time of the day. Thank you.